Hello, bachcho. Let's see the question. So the question says, which vector is commonly used in the transfer of gene in a crop plant? So इन चारों ऑप्शन में से हमें वो organism identify करना है which is used as a vector in gene transfer to plant cells. So the key concept in this question is to understand the process of gene transfer in plant cells. So the gene transfer process in plant cells was first discovered due to the phenomenon which was first seen in Agrobacterium tumefaciens. So Agrobacterium tumefaciens is a type of bacteria which infects plants like sunflower, cotton, tomato, and other crop plants and forms a disease which is known as the crown gall disease. So, in crown gall disease, it results in the formation of tumor in plant cells, especially in the stems of the plants. So the disease causing agent of the Agrobacterium tumefaciens is the presence of the Ti plasmid present in these bacteria. So the Ti plasmid, known as the tumor inducing plasmid, has basically two regions which causes the crown gall disease. So the first one is the tDNA region. So this entire part of the plasmid is the tDNA region. So the tDNA, also known as the transfer DNA, is the tumor inducing region of the plasmid. So this transfer DNA is the region that is transferred from the bacteria to the plant cells. So once this tDNA is transferred from the bacteria into the plant cells, this tDNA incorporates into the plant cells host genome and results in the formation of tumors in the plants due to the tumor inducing genes present in the tDNA region. Now the second part present in the Ti plasmid, which is important for the transformation to take place, is the virulence region. So this virulence region, also known as the Vir region. So this Vir region is responsible for the transformation process. That is, the tDNA is excised from the plasmid and is transformed into the plant cell genome. So this is how the Agrobacterium tumefaciens causes tumor in plant cells. Now, how does this tumor benefit the bacteria? So the tumor cells present in the plants releases factors which are known as opines. Now these opines provide nutrition for bacterial growth. So when the tumor is formed in the plant cells, the bacterial growth is induced in the plant cells. Therefore, the agrobacterium thrive in tumor cells. So this is how the infection of agrobacterium into the plant cells helps the bacteria. Now, how can we use this phenomenon to our own need? So now we can use this Ti plasmid as a vector for gene transfer. So how can we do that? So we can replace the tumor inducing gene present in the Ti-plasmid, that is particularly in the tDNA region, with the gene of our interest. So the agrobacterium that we will get will be a recombinant bacteria. So it will be known as the recombinant bacteria because it will have a plasmid which has the tDNA, but the tDNA, the tumor inducing region of the tDNA has been replaced with the gene of our interest. Now, this recombinant agrobacterium can be used to infect the plant species. Now, the plants will get infected by this bacteria, but tumor will not be formed in that case because the tumor inducing gene has been replaced with the gene of our interest. So, instead of tumor formation, there will be the formation of the proteins of our own interest. So the gene of our interest will be incorporated into the plant cells because the tDNA is transferred from the agrobacterium into the plant cells due to the presence of the virulence region in the Ti-plasmid. And instead of causing tumors in the plant cells, the plants will now produce the proteins of our interest. So this is how we can use gene transfer in case of plants with the help of Ti-plasmid of Agrobacterium. Now, since Agrobacterium only affect plant cells, therefore this Ti-plasmid can be used as a vector in only cases of plants and not in animals or any other microbes. So this is how specifically Ti-plasmid of Agrobacterium is used for gene transfer in the crop plants. So now if we see the options, the first option says plasmid of B subtilis. So this is not used in case of crop plants. So this won't be our answer. Bacteriophages are particularly used for gene transfer in cases of bacteria. So for bacteria it is used. Again, this won't be our answer. Ti-plasmid of Agrobacterium, as we studied right now, will be the answer because Ti-plasmid is used for gene transfer in plants. E. coli phages are used for gene transfer in E. coli only. So this again won't be the answer. So our final answer for this question is option 3, Ti-plasmid in Agrobacterium is used for gene transfer in plants. Hope you understood this well. Best of luck. Thank you.